Good morning, viewer. Welcome to our FTV English version. Viewers, hypertension can begin harming blood vessels, brain cells, and white matter long before a rise in blood pressure is detected, according to a new preclinical investigation from researchers at World Coronary Medicine. And these early effects may help clarify why hypertension is strongly linked to cognitive disorders such as vascular cognitive impairment and Alzheimer's diseases. The study published in Neuron shows that hypertension may trigger early shifts in gene activity within individual brain cells, setting the stage for problems with memory and thinking. And this line of research could guide the development of treatments that not only lower blood pressure but also help preserve cognitive function. Although patients with hypertension faces a 1.2 to 1.5 fold higher likelihood of developing cognitive disorders compared with those without the conditions, the underlying reasons has remained unclear. Many widely used treatments effectively control blood pressure but have limited influence on brain health. The new findings suggest that structural and functional changes in blood vessels may contribute to damage that occurs separately from elevated pressure itself. We found that the major cells responsible for cognitive impairment were affected just 3 days after inducing hypertension in mice. Before blood pressure increased, said senior author Dr. Costantino Ladecola, director of the Family Brain and Mind Research Institute and professor of neurosciences, and Anne Paris Tet professor of neurology and at Cornell. The bottom line is something beyond the dysregulation of blood pressure is involved. Dr. Anthony Pachel, postdoctoral associate in neuroscience at Weill Cornell College, Dubai. Earlier research from Dr. Ladekola's group showed that hypertension broadly disrupts how neurons operate, but advances in single cell analysis have now given the team a way to examine the specific molecular changes occurring within individual brain cell types. To create hypertension in mice, the scientists used the hormone angiotensin, which raises blood pressure in a way similar to human physiology. Then they examined how various brain cells responded after 3 days before blood pressure increased and again after 42 days when blood pressure was high and cognitive changes had appeared. And at day 3, gene expressions dramatically changed in 3 cell types: endothelial cells, interneurons, and oligodendrocytes. Endothelial cells, which align the internal surface of blood vessel, aged prematurely with a lower energy metabolisms and higher sen- sense markers. The researchers also observed early signs of weakened brain barrier, which regulates the influx of nutrients into the brain and keeps out harmful molecules. Interneurons, brain cells that regulate the balance of excitatory and inhibitory nerve signal, were damaged, leading to an imbalance between inhibition and excitation like that seen in Alzheimer's diseases. In addition, oligodendrocytes that undergo nerve fibers with myelin did not properly express genes responsible for their maintenance and replacement. Without enough oligodendrocytes to maintain the health of the myelin sheath, neurons eventually lose the ability to communicate with each other, which is critical for cognitive function. Even more gene expression changes were observed at day 42, coinciding with cognitive decline. Beyond this exclusive report, FTV English, Delhi, Bharat.